Good morning, evening, fellow pre-meds. Morning, maybe if you're living in the UK, fellow pre-meds. Today we have more Anki cards to go through, and actually I did some this morning, and I did 639 cards in 156 minutes, which is really, really, really good. Actually, might be a new record for me. We're 7 days in our streak. Longest has actually been 12 days, and we're just gonna break this record number of cards I've done. We'll probably get aiming for like 800 today or something. So we'll go way and above and beyond our previous record. I still have to do psychology, but I'll do it tomorrow. So with all that said and done, we'll jump right into chapter 3. And this one's actually kind of hard. So I'm probably going to be, just trying to remember the cards and not really recite the information. Enzyme-linked receptors. Enzyme-linked receptors have 3 domains. We first have the transmembrane domain, which is embedded in the plasma membrane. And we have the ligand binding domain, and then we have the catalytic domain. And these enzyme link receptors typically link to secondary or precede secondary signaling cascades or secondary messenger cascades. Non-enzymatic protein. And here's 5. The mnemonic we use is that some motorcyclists bind to immune markers. So we have the S is a signaling protein, I think. B is binding protein, biosignaling protein, immunoglobulins, and some bicyclers bind. Or some motorcycle. We have motor proteins, and I forgot the S. Yes, might be structural proteins. Motor proteins, binding proteins, immunoglobulins, and biosignaling proteins. Primary structures of large proteins, we typically use specific enzymes. So we have this, I think it's like bromium cyanide thing that detects methionine, the carboxy end of the methionine. We also have trypsin, which detects the carboxy end of basic amino acids like lysine and histidine. And we also have chymotrypsin, which detects the carboxy end of aromatic amino acids. So G protein coupled receptors are involved in cell signaling, and they are a family of integral memory proteins. So they have a protein that extends to the inside and the outside of the cell. And I think it's all I remember. Oh, yeah, the 7 cellular transduction, and they have 7 membranes spanning alpha helices. That's what characterizes them. Native page is sort of an amino acid electrophoresis, except it separates based on charge and size. And what it does is that it actually keeps the protein alive and functional towards the end, so you're able to isolate the protein with it still being functional. And the main reason why researchers use native page is that if you're able to keep one variable constant, you can compare the other variables relative to each other. So, if, for example, you have 3 amino acids and they all have the same mass, you can run them on a native page and then determine their relative charges to each other, or vice versa. Native page is when you place protein in page on sharing electric field. No modification is made. The protein will migrate according to size and charge. Yep. Yeah, that's a problem. Proteins are not modified, therefore retain their function. It can be reckoned, right? And on protein similar in size and charge, but then you separate them based on similar characteristics. Yep, sick. Actin. Actins are polymer or made up of microtubules, and actins tend to interact with myosin to generate muscle movement, and formation of actin rings is important for division. Actin has a positive and negative side, which allows more proteins. This is why it's important. Positive and negative more proteins travel unidirectionally along actin filament like a one-way street. Thin filaments and myofibrils too. Okay. Column chromatography. This is where you have a column and you have a stationary phase, and a polar phase, or a stationary phase and mobile phase. A mobile phase elute faster than stationary phase, or rather, molecules that have a strong affinity to the mobile phase will elute faster than molecules that have a strong affinity to the stationary phase. Okay. I forgot the three main proteins: ion channels, enzyme link receptors, and G protein couple receptors. Right. Ion channels, enzyme link receptors, G protein couple receptors. 5 major classes. Some motorcycles bind to immune bicyclers. Structural protein, motor protein, immunoglobulins, binding protein, and biosignaling proteins. G protein couple receptors, a family of integral membrane proteins, and they're characterized by 7 alpha helices that span out the extracellular matrix. Actin are polymers, or they're made up of microtubules, and they form the thin part of the myofibrils, most abundant, I think, cell skeleton structures in the cell, but that's irrelevant. And actin has a positive and negative end, which allows more proteins to move in one direction, unidirectionally, or in other words, a one-way street. So biosignaling is how cells receive and send out signals. And the 3 main types of protein that are responsible for it are the ion channels, G protein couple receptors, and the enzymatic proteins. Enzymatic proteins, or catalytic proteins, enzyme linked receptors. That's the name. Enzyme linked receptors. Dynein. I think I remember. Yeah, it's involved in motor function. One of the 2 motor proteins interact with microtubules, involved in the sliding movement of cilium flagella and transport of cargo towards the negative end of a microtube. So lots of information there. So remember, it interacts with microtubules, involved in the sliding movement of cilia and flagella, and transports cargo and vesicle through the negative end of the microtubule. Migration velocity of a protein. V equals EZ, where E is electric field, Z is charge, and F is frictional coefficient. And you can just make relations with that. So V equals EZF. EZF. It's very easy to get an F in biology, or in biochemistry, which is this chapter. V equals EZF. Z is a charge, E is electric field, F is a frictional coefficient. Okay. And V is the velocity. Keratin, it's part of the intermediate filament, I know that. Intermediate filament contributes to integrity of the cell and functions as a regulatory protein. Primary protein, hair and nails. Mechanical integrity of the cell and functions as a regulatory protein. Intermediate filament epithelial cells. Mechanical integrity and primary protein, hair and nails. Okay. Biosignaling is how cells send and receive signals. 3 main types are ion channels, G protein couple receptors, and enzyme link receptors. Dynein is one of the motor proteins. It interacts with microtubules, and dynein moves and transports cargoes around microtubules towards the negative end of the protein, or towards the negative end of the microtubule. I'm missing one more. I forgot what it was. Sliding movements of cilia and flagella. Okay. Bradford protein assay. Mixes a protein in solution with brilliant blue dye. The dye is protein in green and brown color before mixed with protein. Dye will give up its proton by okay. I don't quite understand this. So next is a protein solution with blue dye that is protonated in green and brown color before mixed with. Okay, so more protein correlates with more blue. So mixing protein with a blue dye, and the more protein there are, the more it interacts with the dye and stays blue, and the more absorbance of the chromatography, and it exhibits a wavelength similar to blue. Okay. Kind of get it. So migration velocity is dependent on the equation V equals EZ over F. E is electrical field, Z is a protein charge, and F is the frictional coefficient. Keratin is a protein involved or associated with the intermediate filaments in epithelial cells. Keratin is a protein found in our hair and nails, and keratin is involved in structural integrity and regulation of the cell. Dynin is one of the motor proteins that interact with microtubules, and it's is involved in the movement of cilia and flagella, and also involved in the transportation of vesicle cargo from the positive to negative ends of the microtubules. Yeah. So centrifugation is spinning things down, I guess. Centrifugation is a process of applying centrifugal force to a solution by spinning it, causing solid particles to collect at the bottom of the solution. Okay. Applying centrifugal force to a solution by spinning it and causing solid particles to collect at the bottom of the solution. Bradford protein assay is using a blue dye to stain proteins, so the higher the concentration of protein, the more it absorbs the blue dye or holds onto the blue dye, which increases the absorbance at the blue wavelength. That's kind of what I'm getting out of it. Collagen. I think these are involved in extracellular matrices. Yeah. Makes up most of the extracellular connectivity found throughout the body. Protein. I think the important part here, really, is making up the cellular matrix. Collagen is the protein that makes up most of the extracellular matrix of the connective tissue. The polyacrylamide gel. Standard medium of protein electrophoresis. Porous mixture with solid acts like a CF. So slows down larger particles, while small particles easy to pass, and it's medium for protein electrophoresis. Okay. Mobile phase move, stationary phase stay. Bradford protein assay. Yeah. And UV spectroscopy. Centrifugation is putting a tube and spinning it down using centrifugal force to make solutes sink to the bottom and solid particles sink to the bottom of your tube. Enzyme link receptors are receptors that have a catalytic function when a ligand binds to it. So it has three domains: transmembrane domain, ligand binding domain, and catalytic domain. And this typically leads to a secondary cascade messenger. Collagen is it's secreted in the extracellular matrix by connective tissue. Isoelectric focusing. Electrophoric method that exposes basic and state protein bases are isoelectric ones are normal. They will have a pH gradient where the pH is low, positive charge near the anode, and the pH is high, negative charge near the cathode. pH below its isoelectric protein have a positive charge, right? Cause it to the protein moving time. So separating protein based on its isoelectric point. Okay. So the important thing to pull off here is that we have a pH gradient where it's low on the anode side, which is positive, And we have high pH on the negative side, which is the cathode side. And it separates protein based on the pI. Right? Yeah. Low pH, positive anode. High pH, negative cathode. Separating proteins based on pI. Polyacrylate gel is the standard used for protein gel electrophoresis. It's very porous, so allowing large molecules to be held back and small molecules to move faster down the gel. Two main ways determined. We have the UV spectroscopy and the Bradford protein assay. Activity, no idea. Known reaction with the So monitoring a known reaction with a given comparing it to a standard. Monitoring a known reaction with a given concentration, so comparing it to the standard. 5 major classes of non-enzymatic protein: binding proteins, motor proteins, structural proteins, immunoglobulins, biosignaling proteins. Integrins are groups of proteins. Choose alpha and beta. These genes are important to binding and to communicating with the extracellular matrix and play an important role. And so this can greatly impact cellular function by affecting integrin. Groups of protein that have 2 membrane spanning chain. This is a lot to memorize. Okay, groups of protein have 2 membrane spanning chains referred to as alpha and beta. And these chains are important in binding to and communicating with the extracellular matrix. Important for binding and communicating with the extracellular matrix, and plays an important role in cell signaling and helps white blood cells migrate and attach to cells. So, okay. It's a lot to take in. But alpha beta spanning chains, binding with communicating EC matrix, cell signaling, white blood cells. Isoelectric focusing is using a same thing as the polyacrylate gel, except has a pH gradient where we have low pH on the positive side of the anode and high pH on the negative side of the cathode. And we separate protein based on its isoelectric point. Actin is makes up microtubules, and it's also found in the thin parts of the myofibrils. It's involved in the function of flagella and cilia, and flagella and cilia. And actin is involved in interacting with myosin to generate muscle movement. Oh, that's the other one. It just goes positive, negative side. That was the other one. Actin protein positive, negative side. Actually along actin. G protein couple receptors are classes of integral membrane proteins, and they have 7 alpha helices that span out. That defines it. You typically give a specific amount concentration of substrate for a given reaction and compare it to a standard. Integrins are proteins that are involved in. Is it proteins? Yeah, proteins have 2 membrane spanning chains, elements alpha and beta. These chains are important in binding to communicating with EC matrix, play an important role in cell signaling, and this can greatly impact cellular function, affecting cell division. Helps white blood cells migrate to ligand-gated channels. I know what they are, but I want to describe them. A ligand-gated channel is a channel that's regulated by the binding of specific ligands. 3 subunits of a G protein. So we have the alpha subunit, which carries GDP to the G to and from the G protein. Nearby enzymes catalyze the beta subunit, structural component of the G protein, and the gamma subunit, which is also a structural component. Okay, alpha subunit carries GDP to and from the G protein. Nearby enzymes to catalyze. Okay, so alpha subunit is like a GDP, transforms over to GTP. Alpha, beta, gamma. Bay or bag. I think bag's better. Alpha, beta, gamma. Alpha, beta, gamma. No, it's all the Yu-Gi-Oh cards. Alpha, beta, gamma. Alpha, the magnet warrior. Beta, the gamma war. Alpha, the magnet warrior. Beta, gamma, the magnet warrior. Oh, interesting. Okay. So alpha units is the one that carries GDP to GTP to and from the G protein by enzymes catalyzed, and the other two are structural components of G protein. Okay. Actin. Actin makes up microtubules, and they're the thin parts of myofibrils. They are involved in interaction with muscles to generate movement, and they also allow motor proteins to move from positive to negative ends, or they contain polarity positive and negative end, which allows motor proteins to move in one direction. Integrins are proteins that have two helices. You have the alpha helix and the beta helix. They're transmembrane, and it's involved in white blood cell. It's involved in cell signaling for sure, and something with extracellular matrix. Alpha chain, beta chain joins the cytoskeleton on the inside of the cell. Okay, I think the important part is highlighted here. Joins the cytoskeleton of the inside of the cell to the extracellular matrix on the outside, and it contains alpha and beta chains. Okay. What is biosignaling? How cells send and receive info. And the three main types of protein responsible for it are the ion channels, enzyme ligand, enzymatic ligand. Oh wait, no. Enzyme ligand channels and G protein couple receptor. Enzyme ligand receptors and G protein couple receptors. Enzyme linked receptors. G protein coupled receptors. Enzyme linked receptors. G protein coupled receptors and ion channels. So I'm going to put as a no. What is the ligand? Ligand channels are protein channels where the binding of ligand changes its conformation to allow it to change, allow for the movement of stuff. G protein has the alpha, beta, gamma subunits. Beta and gamma are the structural components of G protein, while the alpha subunit is something that carries GTP to other enzymes for catalysis and moves it around in a circle. So V equals EZ over F. E is electric field, Z is protein charge, F is frequency or frictional force, and V is the velocity. Keratin is a protein that's involved in with intermediate filaments, and it is keratin provides regulation, structural support, and keratin is also protein found in our hair and nails. But for protein, as we have a certain concentration of protein, and you insert a blue dye, and you measure the absorbance, because the more protein you have, the more absorbance you have for the blue dye. So you measure the absorbance of the blue color. Integrins are proteins that contain alpha chains and beta chains that are transmembranic, and they're the things that actually connect the cytoskeleton of the cell into the extracellular matrix of the cell. Dynin is one of the motor proteins. It is involved in the movement of vesicles from the positive end to the negative end, using motor proteins, and it's also involved in the movement of cilia and flagella. Biosignaling is how cells send and receive signals. The three main proteins involved in is ion channels, enzyme linked receptors, G protein couple receptors, G protein couple receptors, enzyme linked receptors, ion channels. How does one determine? So you determine primary structure of a protein by using certain enzymes. So we have cyanide bromide, which will detect the carboxy end of the methionine. We also have trypsin, which detects the carboxy end of the basic amino acids. And we also have chymotrypsin, which detects the carboxy end of the aromatic amino acids. Oh, okay. Basically, that's sequential and specific digestion of the protein. Okay, using cleavage enzymes, basically. How do the mobile and stationary phase work together to support chromatography? So, stationary phase stay still. They're typically polar, and polar molecules will stay up there and have a longer retention time. Mobile phase are non-polar, so non-polar molecules will have a faster retention time. spinning down a tube and relying on centrifugal force to bring solids down to the bottom of the tube. No idea. Okay, so let's make the sense here. We have a G protein coupled receptor, so we have a G protein that's inactive with an enzyme. So when a ligand binds, the protein receptor's G protein affinity for receptor increases, so it will bind to the receptor using GTP, which turns it to an active state. And it's activating secondary messenger pathways to activate or be inhibited. Okay, so we have a G protein couple receptor here with a G protein that's inactive. And when a ligand binds to the G protein couple receptor, it will cause an increase in this receptor for G protein. And G protein will bind to it by changing it into the active state, causing it to gain GTP. And because it has GTP, it can now dissociate and inactivate or inhibit other enzymes through secondary messenger pathways. Okay, kind of get it. Or two main ways to determine the concentration. We have the UV spectroscopy and Bradford protein assay. Collagen, elastin. This is something that makes up the extracellular matrix of the connective tissue. Polyacrylamide gel is a medium for protein electrophoresis, and it's porous, meaning it allows small molecules to move faster and larger molecules to be left behind, unfortunately. The structure can be determined. X-ray crystallography and NMR. Didn't know that. Kinesin's an our motor protein, I think. Yeah. Kinesin plays a key role in aligning chromosomes during metaphase and depolymerizing microtubules during anaphase. Also transport towards the okay. So kinesin travels toward the positive end. Kinesin is one of the two motor proteins associated with microtubules. Kinesin plays a key role in aligning chromosomes during metaphase and depolymerizing microtubules during anaphase. Also transport cargo to the positive end. So positive end runs on microtubules, aligning chromosomes during metaphase and depolymerizing microtubules during anaphase, and carries cargo towards the positive end of the microtubule. Isoelectric focusing is using a gel, a specific gel, a special gel, and we have a low pH negative charge low pH positive charge anode and a high pH negative charge cathode. And we separate amino acids based on its isoelectric point. Positive charge anode, high pH negative charge cathode. Right. So G protein couple receptor. We first have the inactive G protein receptor. And when a ligand binds to it, it causes it to increase its affinity for G protein. And G protein will bind to it, converting a GDP into a GTP, which activates a G protein coupled receptor, or G protein specifically. And when the ligand leaves, G protein will bind to another enzyme to activate or inhibit it, typically through secondary cascade messengers, secondary messenger pathways. What are ways of structure? So X-ray crystallography and NMR. Kinesin is one of the other motor proteins. It's involved in aligning chromosomes during metaphase and depolymerizing microtubules during anaphase. And it also brings cargo to the positive end of the microtubule. Cell adhesion molecules. Coherins, integrins, and selectin. CAMs are integral membrane proteins. All CAMs are integral. So cell are integral membrane proteins. Oops. There you go. Cadherin, integrin, selectins. They're integral membrane proteins that are found on the surface of most cells and aid in binding a cell to the extracellular matrix or the aerosols. Cadherin, integrin, selectins. Okay. Activity: you use a known concentration of substrate and measure its reaction and comparing it to a standard. Ion exchange chromatography. No idea. The beads, stationary phase, and columns are coded with a charged substance, are negative or positive, based on charge of negative protein of higher affinity, lower retention time. These are code positive ions, negative charge. So using beads that are charged, and based on the charge of the beads, if they're positive, for example, negatively charged amino acids will stay back, while positively charged amino acids will elute faster. Okay. Stationary phase in chromatography is something that it's polar. We have polar beads, silica beads, alumina beads, for example. We have SDS page, native page, and I forgot the other one. Isoelectric focusing. Process of generating electric field separates protein based on their charge and size. Cathodes are negative, anodes are positive. Cell adhesion molecules, or CAMs instead, they are proteins that are involved in keeping cells connected to the extracellular matrix or other cells, and they're usually all integral membrane proteins. Examples are cadherin, integrin, and selectin. Actins are make up microtubules, and they are make up the thin filaments of the myofibrils, and they transport cargo from the the actins themselves are polarized. Some actins have positive ends and negative ends, which allow for unidirectional transport. Ion exchange chromatography is where using the stationary phase is charged. So if a stationary phase is charged positive, for example, and you load a protein that's positively charged, it will elute quicker. As opposed to if you put on a protein that's negatively charged, it will have a longer retention time or elute slower. What method is used to determine the primary structure of small proteins? We can use specific enzymes to cleave specific parts of the proteins to generate or to elucidate the primary sequence. Oh, or Edmond degradation. Okay. So we use Edmond degradation to determine the primary structure for small proteins that are 50 to 70 amino acids in length. We use amino acid N-terminus one at a time and sent to a mass spectrometer to determine their mass and identity. Okay. Electrophoresis is running an electric field to separate protein based on size and charge. Anodes are positive, cathodes are negative, and three main types are native page, SDS page, and isoelectric focusing. Three subunits of G protein: alpha, beta, gamma. Alpha is the subunit that actually moves between GTP and GTB and GDP, activating other enzymes. And beta gamma subunits are the structural components of the G protein themselves. Ligand-gated channels are regulated by ligands binding to the receptor. Biosignaling is how cells receive and send out info. Three main types of it are ion channels, enzyme-linked receptors, G protein-coupled receptors. Common forms of chromatography: size exclusion chromatography, affinity chromatography, size exclusion affinity exchange chromatography, column chromatography. Cool. Tubulin, no idea. Protein that makes up microtubules. Important for brain structure, chromatin separate. I know that. Has polarity. Negative ends closer to nucleus and positive ends using the periphery of the cell. So it makes up microtubules. It has polarity. Negative ends closer to nucleus, positive ends closer to the outside. So for small proteins that are 50 to 70 amino acids in length, we use something called Edmond degradation, which rips off an amino acid from the end terminus one at a time and sends them to a mass spectrometer to identify the amino acid. Integrins are proteins that contain alpha and beta chains, and they are what link the cytoskeleton of the cell to the extracellular matrix of the cell. The three types: GS, GI, GQ. GS, GI, GQ, phospholipase C, which cleaves a phospholipid from the membrane to form PIP2. PIP2 is cleaved from DAS, which is a calcium. Oof, I don't know if you have to know this. GS stimulates adenylate cyclase, which increases level of CAMP. GI inhibits, inhibits adenylate cyclase, which decreases level of CAMP. And GQ activates phospholipase C, which cleaves a phospholipid from a membrane to form PIP2. PIP2 is then cleaved into DAG and IP3, which is a calcium-related secondary messenger. Okay. I don't know if I have to know this. I'll try my best to memorize it, though. Primary structure of a protein. You use specific enzyme to cleave protein in specific parts, and you can elucidate the primary sequence. Tubulin makes up microtubules, and like actin, it has polarity. The negative side is toward the nucleus, and the positive side is toward the outside of the cell, or periphery of the cell. SDS page, no idea. It's used to separate protein based only on masses by adding SDS. So it makes every protein constant because all proteins are more or less the same. Making them depend on mass. Okay, so SDS will disrupt non-covalent bonds in protein, denaturing them. Cause it to revert to primary, denaturing them to primary structures and parts on the protein's massive non-polar groups. Mass the actual charge of protein. These are a constant negative charge. Negative charge. Okay. So it makes it negatively charged. It makes all the proteins negatively charged. I see. Okay. We have GI, GS, and GQ. GI inhibits adenylate cyclase, which lowers CAMP levels. And we have GS, which activates adenylate cyclate, which increases CAMP level. And GQ will inhibit some kind of phospholipid, which involved in some random cascade. Yeah, I want to make sure I get these two. After protein was next step of analysis, no idea. Structure, activity, cooperation, concentration, and composition. I mean, I don't know why I have to know this. Structure, activity, I'll figure out. Ungated channel. No gates and complete. Just holes in the membrane, which are molecule. No gates and basically holes in the membrane, which are molecules faster. Binding proteins, no idea. Proteins with special affinity for specific molecules. For example, there are binding proteins for oxygen called hemoglobin, calcium binding proteins, and DNA binding proteins. Okay. SDS page is where you're using SDS, which does two things. Number one, it makes the amino acids all negatively charged by adding a bunch of nonpolar groups to mask the actual charge of the amino acid. And the second thing is that the SDS will disrupt non-covalent bonds, or in non-covalent interactions, so it disrupts the tertiary structure of proteins, quaternary structure, secondary structure, all the way back to primary structure. So it makes all the amino acids primary structure and negatively charged. Therefore, when you run them on an SDS page, you're basically only separating based on mass. Structure, composition, concentration. I know I have to know this. crystallize the protein, fire X-rays. A film behind catches the rays, and then map and used to determine electron density. Ungated channels are basically holes in the cell membrane. They allow specific molecules to pass through. Binding proteins are proteins that have a strong affinity for certain molecules. For example, there are oxygen binding proteins known as hemoglobins, DNA binding proteins, calcium binding proteins, etc. Kinesin is one of the other motor proteins in that it transports, unlike dynin, it actually transports vesicles to the positive end of the actin filament. And kinesin's also involved in rearranging the chromosomes on the metaphase plate during metaphase, and also depolymerizing microtubules during anaphase. X-ray crystallography and NMR. So we have a G protein coupled receptor that's inactive, a ligand binds to it, and G protein switches from GDP to GTP, binding to G protein coupled receptors. Ligand leaves G protein leaves the receptors and binds to other enzymes, activating a secondary messenger cascade. You crystallize a protein, then shine X-rays, rays through it, and then you have a film behind that catches the electron density and use it to generate a model. Voltage gated channels, no idea. Voltage channel is a gate that is regulated by the membrane potential changes near the channel. What is voltage gated channel? Is a gate that is regulated by the membrane potential near the channel. Membrane potential. Channels, that's a gate that's regulated by the membrane potential changes near the channel. What are cadherins? Cadherin. They're part of the integral membrane proteins. I know that. Group of glycoproteins that mediate calcium-independent cell adhesion. Oh, okay. That's what cadherin does. They hold epithelial cells. Interesting. Groups of glycoproteins that mediate calcium-dependent cell adhesion. They hold similar cells together. Okay. Groups of glycoproteins that mediate calcium-dependent cell adhesion. And cadherins usually hold similar cell types together. What is stationary phase of chromatography? It's the silica bead or alumina bead, and they're polar, and they don't move. They allow for cell adhesion of all integral membrane proteins. They allow cells to stick to the extracellular matrix or to other cells. The three types are cadherins, integrins, and selectins. Isolation techniques, electrophoresis, and chromatography. Ion exchange chromatography is where you use the stationary phase as a charged stationary phase molecule, and if it's positively charged and you put for example, a positively charged protein, it's going to elute pretty quickly. Put a negatively charged protein, it's going to have a longer retention time. And if you have strong affinity, for example, these positive negative electrostatic interactions, you can run a salt solution through it to wash away those other stationary phased proteins. Voltage gated channels are channels that are gates that are regulated by the change in membrane potentials. Ion channels are ion channels proteins that create specific charged molecules. Facilitate diffusion, allow for facilitated diffusion of charged particles. Ungated, voltage gated, line gated. Cadherins are integral membrane proteins that are involved in cells that allow for cells to stick to very similar cells, and cadherins allow cells to stick to similar cells. Groups of glycoproteins that mediate cell calcium cell dependent cell adhesion. Chromatography and what was the other one? Chromatography and electrophoresis. Neutralization, clumping together, or opsonization. Opsonization, agglutination, or neutralization. What is gel electro? What is electro? Running electric field to separate based on size and charge of a protein. Cathodes are negative, anodes are positive. Native page, SDS page, and isoelectric focusing. Column chromatography, size exclusion chromatography, ion exchange chromatography, and affinity chromatography. Ion channels are allowed for the facilitated diffusion of ions. There's ligand-gated channels, voltage-gated channels, and ligand-gated, voltage-gated, ungated channels? Yeah, ungated channels. Cadherins are proteins that glycoproteins that are involved in calcium-dependent cell adhesion, and they typically allow similar cells to adhere together. So, for example, each cadherin allows for epithelial cells to stick together. An antibody is something that binds to an antigen and disrupts it. Yeah, they come from B cells. I don't know why I'm getting to that. Antibody is an immunoglobulin, for example, a protein produced by B cells that function to utilize the immune system targeting good cells. So opsonization, agglutination, and neutralization. So the way UV spectroscopy works is that every protein has a certain energy of light it absorbs. So the more protein you have, the more light you absorb. So let's say we have a tryptophan protein, a single amino acid protein, and absorbs blue light. And we run through it through a UV spectroscopy. We see a big absorbance of blue light, which tells us that there's lots of tryptophan, for example. What is method is used to determine the primary structure of small proteins. So proteins that are 50 to 70 amino acids. In we use something called Edman degradation, which rips off amino acid one at a time from the N terminus and then runs them through a mass spectrometer that allows us to identify it. Antibodies are things that bind to antigen and marks it for destruction from the neutralizes it, recruits our cells to it. Structural proteins. Making up cytoskeleton, extracellular matrix, connective tissue. Partitioning, separation of components by nature of the relative affinities for an initial phase. Separation of components by nature of their relative affinities for the stationary phase. Tubulin is part of the microtubule. Tubulin also has a positive negative ends. The negative ends towards the nucleus, and the positive end is actually towards the corner of the cell. And tubulin, I think that's all I know about it. Yeah. How does UV spec allows us to concentrate? So every protein it absorbs a certain energy of light, and we can run through a UV spectroscopy, and we see a drastic increase of a specific wavelength of light. For example, blue. It tells us that that's a certain amino acid that's abundantly present in that sample. Selectins are integral membrane proteins. Maintains a skeleton, extracellular matrix, and partitioning is determining separating compounds based on their relative affinities for the stationary phase. Facilitated diffusion, moving through a protein, receptor, or channel. Large polar charged molecule. Homogenization. Tissue of interest is crushed into an evenly mixed solution. Myosin interacts with actin. Primary motor protein access to filaments and myofibrils. Primary motor protein interacts with actin and take filaments and myofibrils. We have GI, GS, GQ. GI inhibits cyclin adenylase. adenylyl cyclase, which decreases CAMP. S activates activates adenylyl cyclase, which increases the level of CAMP. Structure, concentration, activity. Selectins they link the carbohydrates of other cell molecules to the cell, and they're found on the surface. SDS page. SDS page is basically using SDS to do two things. First thing is that it disrupts non-covalent interaction between the protein, it denatures them into primary structures, and it also masks the charge of any protein because it makes them negatively charged. All the proteins negatively charged. I mean, run them through the SDS page. You're only separating them based on their masses. Facilitated diffusion is separating large polar and charged molecule through the use of channels. Homogenization is crushing a piece of tissue into an even mixture of solution to typically use for protein isolation. Myosin is a monoprotein that interacts with actin, and it's also found in the thick part of the myofibril. Binding proteins. Special affinity for specific user apparent, for example, hemoglobin's binding protein. Affinity chromatography. Affinity chromatography is what? Beads are coated with a receptor. How long has this been going for? I think this has been an hour, actually. We'll just go anyway. In this method, the beads are coated with a receptor of an antibody that only binds to a specific protein. When a mixture of proteins is poor, so the beads coated with a receptor antibody that binds to space. So when you pour a mixture of protein, only the protein that matches receptor antibody will be stuck in the column where the proteins elute out. Okay. Ungated channels are simply channels that are basically open in the membrane, which allows free flow of large charged polar molecules. Chromatography. Separation of molecules based on their affinity for stationary phase, multiple phase. Binding proteins are proteins that bind to a specific molecule of interest with high affinity. For example, hemoglobin is an oxygen binding protein. There's DNA binding proteins, etc. Affinity chromatography is using receptors or antibodies that are specific towards a protein. And when you pour a mixture of protein, that specific protein that is recognized by the antigen will remain there, higher retention time, while the other proteins will elute out. X-ray crystallography is crystallizing a protein and shining X-rays through it, and the X-rays will go onto a film, and use that to draw an electron density and determine the 3D model of the protein. Voltage gated channels are basically channels that are the gates are determined by, or the state or the status of the gate is determined by the membrane potential near the channel. Ion channels. There's ungated channels, voltage gated channels, ligand activated channels. Basically allow for the facilitated diffusion of ions. Cadherins are They're glycoproteins that recognizes that are involved in calcium-dependent cell adhesion. So they typically bind other cells that are very similar. So ecadherin binds epithelial cells. Isolation techniques of proteins, electrophoresis, and chromatography. Opsonization, agglutination, and neutralization. Antibodies are produced from beta cells, and antibodies recognize antigens and recruit other immune cells to attack it. Certain proteins absorb certain wavelengths of light very well. So, for example, we have a protein that's just tryptophan, and tryptophan absorbs blue light. When we put it through a UV vis, we see a spike, a lot of absorptions in blue light. We know that there's lots of tryptophan in the protein sample. Role of structural proteins, cytoskeleton, connective tissue, tendons, ligaments, etc. Facilitated diffusion is the passive diffusion of large polar charged molecules through protein channels. Partitioning is separating of compounds based on their relative affinity for the stationary phase. Homogenization is crushing a piece of tissue into a small mixture typically used for isolation or analyzation of the protein. Myosin is a mode of protein that interacts with actin filaments, and it's the thick part of the myofibril. Chromatography is something used to separate, contains stationary phase and polar phase to separate compounds. Lectins are glycoproteins, or proteins, intramembrane proteins that recognize carbohydrates of other cells. Affinity chromatography using receptors are proteins that recognize a specific protein. When you pour a mixture of protein, the selected protein will remain bound while the other proteins will elute out. Binding proteins are proteins that bind to a specific molecule with high affinity, such as a hemoglobin, which is the oxygen binding protein, or DNA binding protein. Cool. All right, let's see. We're at 800 cards in 209. That's wonderful. All right, guys. That's pretty much it. It'll take forever, and I'll see you next time.